Back on early Earth, the conditions weren't the same as they are now. For instance, there was no oxygen gas until something called the Great Oxygenation Event occurred. Instead, there were reducing compounds like methane, ammonia, and hydrogen. However, scientists noticed that RNA could have possibly survived in these conditions and carried out processes that allude to their potential of being some of the first genetic material. This hypothesis is referred to as the RNA world hypothesis, which claims that the self-replication of RNA molecules could have driven the evolution of hereditary processes of the protocells. This hypothesis is strengthened by the Miller-Urey experiment, which used a gas chamber to recreate the believed early Earth conditions. The simulation of an ancient water cycle produced amino acids and nucleotides, which are the building blocks of RNA and DNA. Before telling you more, my name is Joshua.
I'm Jayanna Baptiste.
I'm Lexi Ray.
Hi, I'm Saugat Bhandari.
Today we'll explore RNA evolution, a theory that suggests RNA was one of life's earliest building blocks, even preceding DNA and proteins. According to this theory, RNA was the first molecule able to store genetic information and catalyze reactions within primitive cells. This unique role gave RNA a dual purpose, essentially managing both genetic data and cellular chemistry. An important part of RNA's role is how it replicates. RNA polymerases, the enzymes that copy RNA, sometimes make mistakes when copying nucleotides. However, these errors are less critical than in DNA replication because RNA polymerases have a proofreading function. They can back up and remove incorrect ribonucleotides. Just like in larger evolutionary processes, RNA evolution happens through cycles of replication, mutation, and natural selection. These cycles drive RNA to adapt and improve its functions over generations, giving us insight into the earlier stages of life. When looking into RNA evolution, there are two aspects that are pretty important. First, catalytic activity. Some RNA molecules known as ribosomes have catalytic properties. This means they can speed up chemical reactions in cells, helping essential processes move faster. Over time, ribosomes can evolve to become even better at these tasks. The next key aspect is heritability. RNA can store genetic information, so when mutations occur, any beneficial changes can be passed down to future generations. This ability to pass on improvements is crucial for DNA to evolve and adapt over time, driving progress in early life. The evolution and world of RNA works in complex ways, but there are some who have dedicated their time to replicate and research the complexities of RNA to provide evidence for hypothesis. In the study titled RNA-catalyzed evolution of catalytic RNA, researchers set out to demonstrate how RNA can drive its own evolution by using an RNA molecule called a polymerase ribosome to copy and improve a hammerhead ribosome. The study aimed to see if RNA could store and pass on useful information in an evolving system.
The study looked closely at ribosomes, special RNA molecules that help other RNA molecules change. Two types of polymerase ribosomes, 52-2 and 71-89, were tested to see how they could help the hammerhead ribosome evolve. The 71-89 ribosome kept functioning well and formed clusters of active sequences, while 52-2 became less effective. They found that the best new ribosome versions, sequence 3, 5, and 15, performed 4 to 9 times better than the original sequence, sequence 0. A chart in figure 1.b shows how these ribosome sequences evolved over time, gaining new abilities. This study highlights the importance of balance in copying accuracy and RNA length for RNA to evolve effectively. Additional evidence for RNA evolution might come from using advanced sequencing technologies to study RNA populations in unique environments like deep-sea volcanic vents. These environments may still harbor ancient forms of life that could shed light on RNA's role in early evolution.
If you recall, the fourth learning objective from section 1.10 in the Biological Principles textbook asks us to explain why RNA is hypothesized to be the first genetic material. Adding to the information we know from the RNA world hypothesis and Miller-Urey experiment, we can look at the information given to us by the study. The study found that certain ribozymes were more or less effective in the replication process of RNA. The results supported the idea of RNA-catalyzed evolution of catalytic RNA, which essentially means RNA can drive its own evolution. Putting together the RNA world hypothesis, the Miller-Urey experiment, and the results of the study, we can answer the learning objective. RNA is hypothesized to be the first genetic material due to the results from the Miller-Urey experiment, which showed that RNA could spontaneously form in the primitive atmosphere as the first genetic material, which we call the RNA world hypothesis. And this is supported because recent research shows RNA catalysts called ribozymes could catalyze further RNA evolution into modern genetic material. However, there are still some uncertainties despite the supporting research. Researchers don't know exactly how the best mutations of RNA evolved. This study found improved sequences, but not a clear explanation of how these sequences developed over others. This is due to the limits that arise in tracking each generation within RNA populations. There's also uncertainty as to what the results from this study mean for early life in Earth history. They suggest that the hypothetical RNA world is possible and even likely, but there is no definitive proof as to how RNA-based life began. Thank you for watching. We hope you enjoyed.